right, now that it is 2 o'clock, welcome everyone. I'm Christine Buckley with the Brain Aneurysm Foundation. And we're happy to have you all join us for our fourth webinar in our annual webinar series. And we're very fortunate today to have with us Vincent Tutino, Ph.D., who's Assistant Professor of Pathology and Neurosurgery at the University of Buffalo. He's a member of the Foundation's Board of Directors, and he's also a past research grant recipient. So we are very fortunate to have Dr. Tutino speak with us a little bit about the basics of aneurysms, the history of aneurysms, which I think people find fascinating because they probably never really thought of, when people really started first talking about them and treating them, as well as the research on brain aneurysms currently, which is definitely a specialty for Vince. So I'm going to kick it off to you. And again, thank you for being with us, and I hope everyone enjoys this.
Thank you for the introduction. Can everybody hear me? Loud and clear? Okay. There's a lot of people here. This is a great turnout. The pressure's on now. Thank you for the introduction. Like Christine said, I'm coming to you guys from sunny yet chilly Buffalo, New York today, to talk about kind of the history of brain aneurysms and kind of where we are at on several of the research fronts. Just to kind of preface everything, a disclaimer that I'm not a medical doctor. I am a researcher and a biomedical engineer and chemist by trade. So I know more about the research side of things. So if you have questions related to the clinical perspective, I can try to answer to the best of my ability, but we'll have to get another call where Dr. Ogilvie is on here if there's a lot of clinical questions. Just some disclosures, like Christine said, I'm a board member of the foundation. I'm assistant professor at the university, but I also am heavily involved with some startup companies looking to commercialize technology in the aneurysm space. And so today we're here to talk about kind of aneurysms and how we've gained this knowledge base of what we know about the disease and how it progresses and how to treat it. And so here you see a lot of different representations, both in art and in medical imaging and illustration and photographs of what the brain aneurysm is, right? And these are all kind of capturing different facets or different ways that we look at it and study this complex disease trait. But before we go in and talk about some of the research questions, I'm going to go all the way back to how we even began to know what it is. So just kind of briefly, a little bit of history of how we came to this point. So the earliest concepts of what an aneurysm is, just by its definition of kind of this arterial dilation, really came from, almost around 3000 BC by the founder of Egyptian medicine, whose name was Imhotep. Right? I mean, not the guy you see in the movies with the mummies, but is actually an Egyptian kind of doctor at the time, right? And he is the one who kind of first recorded this observation of vessel swelling, although it's not really called an aneurysm at that time. It would take, 2,000 to 3,000 years later for the Greek physicians to actually come up with this concept and really call it an aneurysm. Right. And, another 2,000 years later for the Italian physicians, this guy, Giovanni Morgani, at University of Padua, to describe, to actually show it in the carotid arteries on autopsy. And these are still not yet actually intracranial aneurysms. They are just dilations of vessels throughout the body. Right. But you can see from the earliest, recordings of it, it took about 5,000 years before we then, started describing aneurysms as we think of them today. And the first description of a subarachnoid hemorrhage, or SAH, was actually described by a doctor in Milan, again around the year 176, on a postmortem, an autopsy, where the, again, carotid artery had ruptured. And so it wasn't then until the 1800s where an English doctor, John Blacall, made the link between the aneurysm rupture and a subarachnoid hemorrhage, and really the first historical case of an aneurysm rupturing causing subarachnoid hemorrhage and leading to someone's death was actually the death of this guy, who's the Swedish Crown Prince Charles August, who died of a ruptured aneurysm while he was riding a horse. Although at the time it was misdiagnosed by a lot of people because his death was attributed to poisoning by a rival political faction called the Gustavans. So that was the first case, but it was also kind of the first case of misdiagnosis. Because at this time, really, there were no diagnostic tools. Everything was done on autopsy. And only at this point, occasionally, people would have symptoms that might be recognized as features of these kind of large swelling arteries, or maybe a hemorrhage, but no treatment existed. So, clinicians at that time had no course of action, right? The first treatments were actually developed in the mid 1700s. This is going back in time a little bit, but they were not actually focused on treating aneurysm. They were focused on treating dilations of the carotid artery, which is not an intracranial artery yet, right? You can see here in this example, this is the person's carotid artery in their neck. And one of the early ways that an aneurysm on the carotid artery was treated was by ligating this artery. And of course, it was very controversial because you're basically cutting and cutting off one of the direct passages of blood into the brain. But it was pioneered by this guy named Mason Cogswell. And it was eventually then adopted to treat aneurysms mostly on the internal carotid artery, which is, just as the vessels enter the brain. And it was adopted by this one guy named Victor Horsley, and used in the late 1800s, although many objected to this because, like I said, it cuts off the blood flow to the brain. One of the guys who objected to it was this guy pictured here named Harvey Cushing, who rather than ligate the artery, wanted to use something called a Silverstone clamp, which is pictured here, which you could keep screwing in, and it would reduce the blood flow. So very early on, you could tell that all the clinicians recognized that the flowing of the blood into the aneurysm not only provides, the blood that will then leak out once it's ruptured, but also is probably very closely tied to disease progression. And so you'll see coming up, a lot of the treatments for aneurysm are all focused on reducing blood flow into the aneurysm and cutting the aneurysm off from the rest of the cerebral circulation. But you can see here, this, beginning in the late 1800s, this is when we first kind of entered this surgical era where we realized that neurosurgical procedures would be successful in treating an aneurysm and preventing its future rupture. The best, or the most widely used surgical approach was developed in the 1930s, when that same neurosurgeon Cushing and his resident, who later became another famous neurosurgeon, Norman Dott, they both created what's called the Cushing clip, which everybody on this call probably is familiar with, a clip that goes around the neck or on top of an aneurysm, basically squish it down and eliminate the ability of the blood to flow into the aneurysm sac. And this guy right here, Walter Dandy, he was the surgeon who successfully performed the first surgical clipping of an intracranial aneurysm in 1937, and really began the age of modern vascular neurosurgery. And this has been kind of like one of the preeminent treatment modalities for much of the 1900s and is even used very widely today. I'll note too that one thing that really aided this treatment was the introduction of that operative microscope, which is a large microscope that enhances the view for the clinician. And basically almost all neurosurgery is done under an operative microscope now. And that happened in 1957. And you can see here's just an example of an aneurysm that is then isolated, and a clip is placed around the neck. And you can see here, the clip has been released and is isolating that aneurysm from the flow from the parent artery. In the second half of the 20th century, we basically saw a large boom in what's called endovascular treatments. Around the 30s, the cerebral angiograms have been started to be used to aid in surgical planning. But as the technology developed into the later 1900s, we're able to use this type of imaging, mostly what's called digital subtraction angiography, to allow us to navigate the vessels and treat aneurysms and other cerebrovascular diseases from within the vessel, which is called endovascular techniques. And so by the 80s, this guy named Guido Guglielmi at the University of Rome developed something that's called, this is the guy right here, detachable coil, which they're kind of demonstrating here with a slinky around the guy's neck. But this is what it really looks like, and it's a complex structure of a coiled coil that then has this tertiary shape you can see here. And what it's designed to do is be deployed from within the vessels into the aneurysm sac, and then eventually electrical current is run through. And at this point here, that current causes this platinum metal part to detach from the guide wire here and eventually leave this as a coil mass within the aneurysm dome. And this is a scaffold then for red blood cells and platelets to then clot around and eventually limit the ability of blood flow to go into that aneurysm, and it eventually clots off the lesion. And so this was successfully deployed in a first patient in 1990. So I was even alive by that. So it doesn't seem that long ago for me. But there's also been other advancements since the endovascular era. There have been other, not necessarily endovascular, but there have been other surgical techniques, some that are called bypass surgeries. So you can see here, this is a drawing of an aneurysm on the, I believe this internal carotid artery that had been coiled, and it's basically almost occluded this whole artery. And so what the clinicians have done is take a vein graft and suture it from the part of the internal carotid artery that is remaining to the rest of the brain circulation here, to provide blood flow to this part of the brain. New endovascular strategies have also been developed and continue to be developed. One of these being the flow diverter. You can see down here at the bottom of my screen. I don't know if you guys can see my mouse, but I'm pointing to it. It's called the pipeline embolization device. And it's basically a stent that is actually a very dense mesh network that is deployed across the aneurysm orifice within the parent artery, and it basically prevents blood from flowing into the aneurysm. There's other devices like the Webb device, which is called an intrasaccular device, you can see in the picture above, where it's essentially the same concept, but it's a device that is deployed within the aneurysm. So it still has this kind of preventative shield at the neck of the aneurysm that's preventing the blood from flowing in, but the device is all within the aneurysm rather than within the parent artery. And there are other treatment modalities as well. Another one being something that's called a liquid embolic, and one example is a liquid called onyx that is injected with a microcatheter into the aneurysm sac, and once it touches water, the water that's in your blood, it begins to solidify. So rather than relying on blood clotting and blood stagnation, it itself solidifies and prevents blood from entering the aneurysm. And so just to kind of recap, since the late 1800s is really when we saw and have recorded these first published literature in scientific journals on brain aneurysm. And these graphs that I'm presenting here are all data that I've downloaded from the National Library of Science that the government maintains. And so you can see, that's when the earliest publications were, but it wasn't really until the 40s and into the 60s where the number of publications every year on this disease really began to take off. And now it's very well known and very well published on in the early 2000s to now. Other things that I haven't really talked about, but there's been lots of clinical research on, include the epidemiology and the demographics of aneurysms, outcomes in healthcare, cost research, and other kind of concurrent pathologies that are associated with aneurysm, like vasospasm and delayed cerebral ischemia. I'm going to kind of talk about some of the common questions that we have in research right now. And the basics, is what are aneurysms, right? And this, like I've kind of shown previously, has been addressed by a lot of the clinical research that has happened, over the past several hundred years. But this is kind of how we understand it right now. So an aneurysm is this pathological outpouching of a cerebral vessel that mainly occurs on vessels within what's called your circle of Willis. So that's this network, this redundant network of blood vessels that sits at the base of your brain, that in your skull, and it's fed by your internal carotid arteries and your basilar artery that comes up by your neck. And aneurysms are typically found in several different locations within this circle, but can be found outside, as shown here, in this vertebral artery, or not quite vertebral artery aneurysm at the bottom. They generally form because of something called degenerative remodeling. So it's not like you're blowing up a balloon and the blood pressure causes your vessels to push outward. It's actually an active, biologically driven process where the tissue is remodeling outwards. And it's almost like it is remodeling that way in an uncontrollable fashion. And it's typically associated with a high amount of inflammation. And this is prevalent in about 3 to 6% of the US population, but is almost double that in certain groups like the Finnish and Japanese populations. They're typically detected at ages between 35 and 60, and is associated with several risk factors, including smoking, several genetic disorders, family history, and hypertension. and only a small fraction of them are symptomatic. Most of them are asymptomatic, and most are actually discovered incidentally, meaning that they're discovered almost on accident when a person is having medical imaging for other reasons, right? And why they are so important for us to study is because they can rupture and they cause what's called a non-traumatic subarachnoid hemorrhage, meaning that it is bleeding into the brain that is not the result of trauma, like a car accident or some penetrative wound. And about 30,000 people in the US experience rupture every year. That's generally about 1% of all the aneurysms will rupture every year. And that is kind of worldwide, that's about 500,000 people. There's an extremely high mortality at 1 month. 50% of those experiencing a subarachnoid hemorrhage will die by that time. And the cost of care tends to be astronomical. It's about, in the US, about $3 billion every year to treat those who have a rupture, even though that's only 1% of the aneurysms every year. And the median age of those that experience rupture is 52, meaning that, these people are kind of taken during the prime of their life, when they're still working, when they have a family. So the societal cost is also very high. And it's also irreversible, the hemorrhage, because the treatment options after this are very limited, and there's a high chance of subsequent pathologies like we talked about, like vasospasm. So that's in general what we, some of the demographics and and comorbidities surrounding aneurysm. But how we know about the biology has also kind of gone hand in hand with the surgical sciences. Once we started open skull surgery to treat aneurysms and to clip them, many physicians were able to either take parts or whole aneurysms from those patients when they're surgically repairing them and study their tissue. So beginning in the 1940s, there have been several, 100 studies every year or so that have been published on looking and characterizing aneurysm biology based on human tissue alone. And in the late 1990s, there were several key publications that kind of were seminal works, those by Fruzen et al., Katauka et al., and Chayet et al. And these really helped define the structure and the inflammatory structure of the aneurysm tissue across different phenotypes or different classes of aneurysms. And specifically, they characterize the differences between the ruptured and the unruptured ones, where in the ruptured ones, you were able to see a lot of inflammatory infiltration, a lot of cellular death and remodeling characteristics, and a lot of degeneration of all the tissue, especially at the site of rupture. So that's, about 30 years ago is when we were characterizing these differences on histology. And since then, we've then adopted many molecular techniques to understand the biochemical and physiological processes that are happening in the aneurysm, using things like in situ hybridization and immunohistochemistry to characterize the function of certain proteins that are upregulated in the diseased tissue. And we've been able to identify several key biological processes, like I just kind of alluded to here, inflammatory cell infiltration, oxidative stress, and remodeling and degeneration of the tissue. But really, to answer the question of why aneurysms form and how do they grow and rupture, it has been very hard to do with only human tissue, because these studies and this data here this is just one snapshot of the biology. It's what we call cross-sectional data. This we can only observe at one time point because this is the time point that this tissue is being removed from that individual. So in order for us to understand mechanistically how the aneurysm is forming and growing and can eventually rupture, we have resorted in the field to using animal models, right? And this, is what we've been doing to really get at the biological mechanisms. And so you can see here, the first animal models of aneurysm really began in the 1960s, where a group with White et al. And Hassler et al. Began using rabbits and canines to induce some of the first artificial aneurysms. And most of the models since then describe aneurysm formation as a response to the induction of several risk factors. And here's kind of a timeline that we've put together, to show some of the classic animal models across the last, whatever, I think it's like, 60 years almost now. And so that's what we call an endogenous animal model. And so the most famous one is that by Hashimoto et al., which was introduced in 1978. And it's actually still very widely used today. It involves, if I'm correct, it involves the induction of hypertension, wall weakening by feeding the animals a diet of this compound called BAPN, and it involves a hemodynamic manipulation where one of the arteries is ligated, which causes compensatory high blood flow in the other side of the brain. And so these are also a collection of a lot of the risk factors that are induced in an animal in order for it to kind of naturally grow an aneurysm at different locations in the brain. And you can see here's an example from one of our studies a long time ago where you have, it's just, I believe, blood flow alone. So increasing the blood flow to one part of the brain causes friction on the wall at this specific location, and eventually over several days degrades some of the structural components of the wall, and the rabbit in this case begins to form an aneurysm. And this is kind of the counterpart in the rat in Hashimoto's model. And we can use these to now study the impact of different risk factors, molecules, drugs on the genesis of aneurysms, right? And the other interesting thing it allows us to do is isolate biological phenomena and see how those specific risk factors are involved in the development of aneurysm. And so over time, we've used these strategies to really understand key molecules that are involved in the development of aneurysm. Some of them are listed here. And this has been, one of the ways where we're kind of piece by piece putting together how aneurysms form and the mechanism of this disease. But really, some of the latest developments with animal model work has come in just the last 10 years, where we've actually been able to make the aneurysms grow further to a point which they rupture. And this has been developed by two groups, the Hashimoto group and Brian Ho's group, I guess in the early 2010s. The kind of downside of this is that these models aren't really endogenous. They're not a response to risk factors, because the way that they're formed is you inject an enzyme called elastase, which starts to degrade some of the structural portion of the wall, and it becomes so weak that it can rupture. But it at least enables us to start to study the processes that are going on towards the end of the aneurysm's life cycle, and has given us the potential to begin to test drugs before human trials and all sorts of things. So it's been very helpful, even though it does have kind of the downside of not being, such a natural model. And the last thing that I'm going to talk about is, how we can better detect dangerous aneurysm. This is where a majority of my research interest lies. How we have done this in the past has all been by medical imaging, right? And for the most part, the advances in medical imaging have paced with our knowledge about aneurysms in humans. As you introduced the surgical microscope in the early 1900s, that's when we can look at, what is making up the wall of an aneurysm? What do they look like? What are the phenotypes? And then as you introduce angiography, you can look at the shape and the morphology of aneurysms and where they're located. And so you can see just on the publication record alone, much about of the studies about aneurysms actually involve medical imaging. And a majority of this involves what's called angiography, the visualization of blood vessels. And so there's three types of angiography that are mostly used: digital subtraction angiography, which is the invasive modality where dye is injected and it's X-ray imaging, computed tomography angiography, and medical resonance angiography, which are other imaging modalities. Some can use contrast agents, some don't, but they're less invasive and they don't require catheters, catheterization of your arteries. And so, even today, though, research in angiography continues at the center that I work at, UB, called the Kin and Stroke and Vascular Research Center. We have several PIs in our center that are involved in researching angiography and new methods. Those include high spatial resolution angiographic detectors. So these are X-ray detectors, high temporal resolution detectors, basically looking at high number of frames per second to really visualize the flow of the dye in the aneurysm, tracking of radiation dose, and research into how can we better visualize what we see here, sometimes in this MRA image, sometimes 2D. How can we visualize that in 3 dimensions so that we get a more complete picture? But there have been a lot, especially in over the last 30-40 years, a lot of new imaging modalities that we're continuing to study. Some of them listed here are optical coherence tomography, intravascular ultrasound, so using probes within the vessels to look at the walls of the aneurysms or the walls of the arteries, vessel wall imaging on MRI, looking at how these weakened aneurysm walls uptake certain contrast agents. And then there's also been development of new contrast agents that allow us to visualize different facets of an aneurysm. Like this is USPIO, it stands for ultra small paramagnetic iron oxides. They're compounds that certain inflammatory cells can actually uptake or ingest, and we can visualize where these immune cells are in the aneurysm tissue. So these are all at the cutting edge of imaging studies. And what in the modern day kind of permeates everything is artificial intelligence. So kind of on top of the development of new imaging techniques, we have the development of new ways to analyze the output of angiography that we routinely get. And a lot of these include computational pipelines for automatically detecting aneurysms in medical images, or pipelines for segmenting medical images for better visualization, or even mapping different information onto the blood vessels here, such as blood flow information or vessel enhancement intensity. And this is an example of some of this here. In the 90s, we sequenced the first human genome. And since then, there have been other, there's been research into how we can detect aneurysms from a genomic or transcriptomic standpoint. So we know that genetics and genomics, that there's a large component of that into the risk of having an aneurysm and even the risk of a rupture. And so other research into how to detect aneurysms and how to know which ones are riskier has focused on this kind of new omics research rather than the medical imaging. And so you can see this is, though, very much still in its infancy compared to the rest of the research. But this is what my research focuses a lot into. So we look at, how can we analyze the variants that are in someone's genes that are associated with aneurysm, or how can we look at what their cells are doing in the RNAs that their cells are expressing, to see if we can identify, oh, this person might have an aneurysm. Right. So when it comes to genetics, like I said, sometimes we can look at across huge populations of patients with and without aneurysms to try to find genetic factors. Or like in this example, we can look into single families. Each of these, what are called a pedigree, is a large extended family of people who, have an aneurysm or have had a subarachnoid hemorrhage, and those that don't. And we can look in this very controlled environment, because all of them are genetically related, what things are off in their genetics. And we call these different gene variants. And so that's one way that we might eventually be able to have some sort of blood or saliva test to detect if somebody has an aneurysm. Other ways of doing that are looking at what's called transcriptomics or proteomics, which is basically the genes that a tissue or blood are expressing. And so we can also look at, and that's an indication of what are the biological processes that are ongoing in that patient, in somebody that might have an aneurysm. And so we put all of these together and look at this kind of holistically, and this is what we call, eventually could be a potential biomarker. So we can look at the features from the patient's blood and from imaging and see how the differences in the patients that have aneurysms and controls and those that don't, and see what is different in all these features, and then use these kind of machine learning kind of computational models to input that data and delineate individuals in new cohorts. And so that's kind of where we're at from a very high level of going forward and potentially developing some biomarkers that use a patient's blood. And so, the end goal of a lot of this research is to basically, have a doctor, maybe they order a test in a patient that could have familial history. The blood is drawn, sent to a lab where it is then, analyzed with this machine learning classification algorithm. And that report is sent to the doctor to make a subsequent decision of, does this person need imaging? And then we could potentially detect an aneurysm, or do they not, right? And so with that, I'm going to conclude. Here's a list of references that I presented on, and some other interesting resources. And with that, I want to say thank you and ask if anybody has any questions.
Okay, thank you, Vince. That was fabulous. Really appreciate you taking your time and sharing all that great information. You can keep an eye on the chat, but I had asked for people when they registered to send some questions, so I'm gonna go through that list for you now. Somebody asked, how long before we fully understand the genetics of aneurysms? Are we more or less than a decade away? And are you working with people internationally?
Okay, that's a good question. So that's a very good question. I think we're probably more than a decade away from fully understanding, right? So when you look at some of these large genetic studies, like I was describing, called GWAS, right? We can identify what are called SNPs, or single nucleotide polymorphisms, that are associated with aneurysm, but that doesn't actually get us to the actual variant that is causing, that has some causal relationship to the development of aneurysm. And really that's the next step in a lot of the genetic research, is understanding what are these causal variants, and how they actually influence disease progression, right? So they might influence disease by totally knocking out the one gene in its production of a protein, but it could also be creating instability in a promoter or an enhancer region, which is part of gene expression, right? And so until we know what these variants are and what they're doing, then we can really truly understand more the genetics. But that's I think with the advent of things like CRISPR, where we can start doing gene editing and in the lab test these out ourselves, that's one technology that is really gonna advance the field.
And I was reading through other questions. Did you mention how you work with other people internationally? Because I know you work with other centers in the US and Canada, but you work collaboratively with other
Yes,
folks.
I'm in touch with some of the groups in Switzerland. We've been trying to share some genetic information, although my research specifically is more focused on the transcriptomics part. But we have had some international partners on this. Yes.
Okay. Great. Another question, how can someone get enrolled or be a part of a study or join a research group, just in general?
Let's see. Well, if they're interested, they can reach out to me.
Yeah.
There's some projects that we have coming up in the future where we're going across some different centers in the US, where we're going to be collecting samples from individuals. Otherwise, contacting kind of, in your local hospital if they're doing research. But if they get in touch with me, I can see like if I know anybody in their area who's actively doing this kind of thing.
Okay, maybe if you have a second, if you want to put your contact or email something in the chat, people
Yeah.
that. Let's see. Someone's asking, is there a way, and I think there is, from my limited understanding when I see all our research grant applications come in, but are there ways to find weaknesses in blood vessel walls to understand that they may or may not develop into an aneurysm?
So yeah, I think there's some imaging technologies where we're trying to look and understand the weakness of the vessel wall. And one of those things that I do is called vessel wall imaging. And basically what you do is you take a contrast agent, you inject it into the patient, and after a washout period, you image them. And wherever this contrast agent is taken up in the vessels, it will have higher intensity on this type of MRI. And so the thought is that the regions of the aneurysm wall that are uptaking this are weaker. But the issue is we don't know why they're weaker. They could have something like the degradation of these kind of junctions between the cells, which allows that contrast agent to permeate into the wall. But they could also have other mechanisms by which that contrast is actually localized there. So we are in the process of researching, why do some aneurysms have enhancement and others don't, and if this is truly associated with inflammation and the weakening of the wall, then it's a very powerful imaging biomarker.
Okay. Another question, let's see if you have an answer for this. Are you aware of any research on memory deficit progression for survivors? Is anyone looking at, on that rehab side, anything that is relevant to memory loss consistently with someone post-rupture?
I directly don't know. I recently reviewed a paper on something very similar, so I know that there is research out there related to this, but I personally don't know of
Okay.
anyone who's doing this, but I'm sure there is out there.
Okay, thanks. And then someone wants to know, can neurons connect in the area where aneurysms occur?
Can the neurons? Well, theoretically, they are already have connections there. The neurons. The aneurysm is outside of the brain tissue. It is not, part of the brain tissue. So it's not impeding neuron growth or regeneration. But certainly when the aneurysm ruptures, then it does cause tissue damage. And if that's what he's talking about, again, I'm not, like I said, I'm not a medical doctor. So this is not, the brain is not my area of expertise. The vessels are.
Yeah.
But we do know that the brain and the vessels are separate. So I wouldn't see why not.
Okay, and then someone
Answer
wants,
the question.
it's not easy. What can be done? Someone's just asking for more research in order to promote early detection and prevention. Like, what can people do to help elevate research that's being done for this disease?
Well, you can get in touch with the Brain Aneurysm Foundation to promote Ellie's Law, which is a bill that's in the Congress and Senate to appropriate money to the NIH and basically direct them to fund more aneurysm research. Yeah, that is probably one of the best ways.
Yeah, and just to add to that, I mean, in seriousness, I mean, our foundation is the largest private funder of brain aneurysm research, and we've been working really hard up on the Hill to get this extra $50 million via Ellie's Law. But as Vince knows, our funding comes from people who, just donate to us, who are philanthropic and have events for us. So any funding that Vince's studies have had in the past have come through relationships that we have and people who are passionate about the disease and engage with the organization. So we certainly always encourage people to do that. Let's see. And then is there any, I think you alluded to maybe to this a bit before, is there any demographic difference between European and Asian people related to aneurysm prevalence?
So are they referring to the Finnish and Japanese populations, or just European and Asians in general?
That's just what I'm reading the exact question. So, I mean, I think just how does it differ maybe from other continents or?
Well, I think that comes down to the genetics, right? And it's not really well known, but I think there have been a lot of publications on the genetics in certain Asian populations, and certainly in the Finnish and in the Japanese populations. So it likely comes down to the genetic factors, but I, off the top of my head, I would have to read more about it before answering with even greater specifics. So
Nope, that's fine. And I think you can probably answer this one. I mean, again, just based on, where we funded research, I could add on, but I'm pretty sure you can handle like where in the states are the larger centers who are conducting research for brain aneurysms in whatever space it might be.
Yeah, so in Buffalo, we have a very large stroke center and a very large research center here. But some of the other places are, Mayo Clinic in Michigan, Johns Hopkins. There's groups in Chicago. Definitely, we have collaborators in Duke and a group in Iowa. So a lot of the kind of major research institutions. I'm sure there's lots of other ones that I'm forgetting.
Yeah, no, I mean, there are, I mean, for us, I think it's been certainly Buffalo, Gainesville, LA, UCFS, the Barrow,
Yeah, Barrow. Yeah, I forgot Barrow.
Houston and Texas, Emory. I think a lot of the major centers that treat have a large component of research being done as well. So here's another question, again, back to the support. And again, it says, is there more financial support for research each year, private and public? And I think, again, your perspective, you had to form your own startup and attack getting funding a different way, right? Maybe you can.
Yeah. So I think a lot of the support is public, but it really kind of comes down to the percentage, I guess, for each different type of disease. So the NINDS, which is the Institute for Neurological Disorders and Stroke, they fund a lot of ischemic stroke work, but not really a lot of or not really proportionally a lot of research into aneurysms, which cause hemorrhagic strokes. And so for that reason, like Christine said, I have these startup companies where we have sought to get funding through other grant mechanisms that the government has that is still publicly funded, but it's going towards commercial application, and we've received private funding as well in my lab. So
Okay, thank you for that. This is another topic, a question off topic, but it comes up a bit, and I'm sure you've been asked this quite a bit. Somebody is on here that has a 12-year-old child whose dad died at the age of 30. So they're concerned about when to have this child scanned. So what do you hear out there is the common answer to that?
Yeah, so I think pretty much the common clinical practice is that you have to have 2 or more immediate family members who either had an unruptured aneurysm that's been diagnosed or have had a rupture. And so, that's kind of the benchmark for receiving insurance coverage of a scan. But I think, and a lot of neurosurgeons also think they wish that they could scan more people. And that's kind of, the motivating factor behind a lot of the research that we've done in terms of blood testing is, how do you know that you're not going to be the second one in your family that has one, right? And so where we're at, thinking about blood tests is how can we, even if the sensitivity is not as high as some other diagnostics for rare disease, how can you find the patients that are most likely to have one so that you can then suggest them to go get the definitive diagnosis, which would be the imaging. And so, and we would hope that we can apply this strategy to people that might have only one family member or might have, a group of many different important aneurysm risk factors, but might not even have a family member. But just because you're in that high risk group, maybe you can receive the blood test that could suggest you go get, an MRI or a CTI scan.
Yeah. Okay, thank you. And that might be another endeavor for the foundation up on the hill, and we've had started with some conversations with some congressmen who represent some other diseases where they are working with insurers to promote having a scan. Just, I mean, from our perspective, it's obviously for the lives of people to save and, family units in society, but when you go to the hill, everything becomes about money. So it's proving that cost savings. And there are doctors who are working to show the cost savings of scanning, particularly more at-risk populations, particularly women with high blood pressure and who are smokers or African Americans. So hopefully at some point that will also be something that gets a little more traction and that might actually come into play. One last question for you, Vince, before we wrap up, since we do like to stay timely, and this session will be recorded and we'll have it out via social media to anyone that registered in the next couple of days. But is there less funding for SAH brain aneurysm research than for stroke?
I mean, on a whole, I think there is, right? Just dollar amount. But I think too, like as a percentage of, or proportionate to how many people are afflicted, there also might be less. I think that's something that the foundation has looked at and reported stats on. That it's how many dollars per person?
$2.08 per person affected.
Yeah, so that's how much they're spending on aneurysm research. I don't know how much on other types of other stroke research off the top of my head.
Yeah, that's fine. All right, I think, Vince, any closing remarks?
If anybody has any additional questions, I mean, they can feel free to reach out to me. I put my email in the chat here. So yeah.
All right, well, thank you again so much for sharing and for your time. Thank you for everybody for attending. And please continue to follow the foundation on social media, sign up to get email alerts, and, consider donating for more funding for research. Last year, we gave out $630,000 to 16 different projects. Our research symposium this year will be September 21st in Charleston, South Carolina, and will also be our second day of engagement. So definitely just stay tuned with us, and always reach out to us with any other questions you have as well. All right. Take care, everyone. Have a good rest of the day. Bye-bye.